Oh, hello. Good morning or afternoon, evening. I know there's folks from all around the world joining us on Divestream 875, the remotely operated vehicle Sebastian. We've just touched down about 825 meters below the surface of the ocean. It's going to be a shorter dive, a few hours today, but a lot of life here so far. So we've got to scoot a little ways to our south before we get to our waypoint here for doing some push course potentially. So we're going to scoot and keep an eye along the way for anything we might see. Don't know that we'll stop for long for anything, but we're going to keep our eyes peeled. I'm going to turn up the rest of the mics here in mission control so you can hear the chatter. So enjoy the dive.
So let's have a look at these lights while we're going. That way.
Hydrates, turn off photo iris.
Yeah, turn it off. Yeah. 250 or whatever. So it's a little bit less intense. Probably better, I would say.
Hola a todos, buenos días, buenas tardes, desde donde nos escuchen.
And then like this hole down here.
Mi nombre es Valerie de Anda y estoy aquí en Mission Control con Galen, Buzz, Gabriela y Brett. Como menciona Alex, este va a ser un dive corto, pero bueno, estamos viendo una gran diversidad de peces, una gran cantidad de peces aquí a 800 metros de profundidad y nos estamos dirigiendo hacia nuestro punto de muestreo, en donde vamos a colectar más sedimentos. Esta es una zona bastante productiva. Vemos una gran cantidad de peces muy pequeñitos.
Someday I'm going to have a button. That's the thing. When you're an outreach, literally all I want in life at this point is the button and the lights are chased.
Gracias a todos los que han seguido estas transmisiones, los que han estado al pendientes de estos live streams. Les agradecemos que sigan aquí y bueno, admirando las profundidades del mar. Parece haber muchos juveniles, muchos bebés en esta zona. Les recordamos que el láser que ven aquí, estos dos puntos son 10 centímetros aproximadamente. Aquí los radioescuchas que han seguido también expediciones anteriores han ayudado a identificar la diversidad que hemos visto de invertebrados a profundidades de 2.000 metros, 2.700, 2.800 metros. También diversidad de peces que hemos visto, así que bueno, todos sus comentarios y participación en el chat. Ahora nos vamos a dirigir al punto de muestreo y colectaremos sedimentos a esta profundidad. Y bueno, muchas gracias a todos los que nos escuchan, a todos los radioescuchas que están siempre al pendiente, mostrando su interés y su curiosidad por entender nuestro océano, lo que pasa a estas profundidades. Y bueno, también parte de esto es que sigan su curiosidad hacia el océano, y que invitar también a proteger lo que no conocemos, que es uno de los ecosistemas menos conocidos, el fondo del mar y la diversidad asociada. Hay demasiados peces, muchos juveniles en esta área. Parece una zona muy productiva. Bueno, estamos maravillados aquí viendo esta gran cantidad de peces en esta zona. Le voy a preguntar a Gabriela del ministerio a ver si tiene una respuesta por qué esta zona es tan productiva. Se espera, bueno, aquí en el mapa lo pueden ver, la ubicación. Algunos de ustedes preguntan dónde nos localizamos. pueden ver el link de la página de SOI. Ya estamos en el fondo, no vamos a bajar más. Estamos a 834 metros de profundidad y es aquí donde vamos a tomar las muestras. Nos dirigimos un poco más al noreste, pero será un dive más corto. Bueno, aquí todos estos peces están. Es una zona donde vemos muchos juveniles. Distintas especies. Hemos visto también un calamar al descenso, pero bueno, este será un dive más corto. También vemos algunos ofiurios al fondo, las estrellitas de mar, que bueno, vemos que son bastante sensibles ante el movimiento, se mueven. Es una zona bastante productiva. Vemos igual como en otras live streams la gran cantidad de materia orgánica que caracteriza estas zonas. Es una área de mucha productividad y emocionados de poder analizar los resultados tanto de la superficie de agua, la columna de agua, como los sedimentos, para poder tratar de entender todos los procesos que están ocurriendo en esta área.
If he goes quicker, all these people go straight there.
Alright, Chris, go ahead.
Hey Tom, can we increase speed, please?
Yeah, Chris is 47.
I'm not sure who's talking yet.
Yeah.
Y bueno, les recordamos que pueden hacer sus preguntas. Hemos recibido preguntas también para el equipo de ROV, preguntas acerca del submarino, acerca de la expedición, también hemos tenido preguntas acerca de perseguir carreras científicas, carreras para estudiar el océano, biología marina, oceanografía, curiosidades e inquietudes como parte de este acercamiento de la ciencia a todos ustedes que nos ven, que nos escuchan. Kevin, can you log in page? Log the page.
It's wrong, I think.
It's calibrated.
Yeah, I don't think it's right.
Nos acompaña Ceci, que estaba terminando unos experimentos. Siempre está trabajando hasta altas horas de la noche. Y bueno, Ceci, ¿qué nos puedes decir? Estamos en una zona llena de estos pececitos, muchos juveniles. Una zona de gran producción primaria en la superficie también.
Sí, se ve desde lo vemos acá cuando bajamos con el ROV, lo vemos cuando hacemos los experimentos en que medimos los procesos, y acá ocurren en gran magnitud la producción primaria, la respiración, y también lo vemos con la enorme diversidad de organismos que hay, tanto en la columna de agua como en el fondo, y también en la superficie. Tenemos a Lucía y Gabriel haciendo los avistamientos y están viendo cosas maravillosas. Como por ejemplo hoy, Gabriel había un pez luna saltando afuera del agua. Sí.
Wow.
Y ahí hay muchísimas aves. En este momento están haciendo un censo importante
Hay
de
muchas aves, sí, muchas aves también. Entonces lo vemos a todos los niveles, en la superficie, en la columna de agua, aquí en el fondo.
Para quienes preguntan acerca de la nacionalidad, habemos 4 uruguayos a bordo como parte del equipo científico, que somos Cecilia Alonso es mi nombre, yo soy la coordinadora por Uruguay, y tenemos a Juan Zanetti, Lucía Frones, Danilo Cagliari, y tenemos también a Gabriela Jorge, que es observadora a bordo por el Ministerio de Ambiente. Y después tenemos una tripulación internacional, a Valerie, que ya la conocen, que es mexicana, y colegas de distintas partes del mundo, tanto en cuanto a la tripulación científica como a la tripulación del barco que maneja el barco y el ROV en particular.
Y bueno, esta expedición se llama Buscando las Asgard, en busca de nuestros ancestros, las arqueas Asgard. Estamos particularmente interesados en este grupo de arqueas que no se conocían, hace 10 años se descubrieron, y estamos interesados en buscar estos organismos en ambientes como estos, ambientes ricos en producción primaria, en materia orgánica, donde sabemos que se han visto en otros sitios parecidos, y tratar de entender cuáles fueron los procesos que dieron lugar a el origen de la complejidad celular. Estas arqueas, los ancestros de estas arqueas estuvieron involucrados en el proceso de eucariogénesis, el proceso que dio lugar a toda la vida que conocemos, a toda la vida y biodiversidad que podemos ver a simple vista, que es como podemos ver aquí todos estos peces, árboles, incluso nosotros los seres humanos. Entonces, entendiendo, conociendo estas arqueas, podemos entender mejor de dónde venimos, cómo se dio lugar el proceso de vida compleja y también sus procesos ecológicos, la importancia que tienen estos organismos en el ambiente. Bueno, además de las Asgard Archaeas, estamos caracterizando muchísimas cosas dentro de esta expedición a nivel la superficie del mar, la columna de agua y los sedimentos, ¿no? Juan ha hablado y Ceci han hablado de los experimentos que están haciendo en la columna de agua, Danilo también, lo que estamos buscando también en los sedimentos, tratar de tener todo este conocimiento para integrarlo de una forma en la que podamos dar un mejor entendimiento de los procesos que están ocurriendo y que pudieron haber ocurrido hace 2.000 millones de años. Aquí hay una pregunta acerca de astrobiología. Si existen marcadores de la Tierra primitiva, si los organismos dejaban marcadores en la Tierra primitiva o en la atmósfera para poder inferir que hay vida. Cuando los microorganismos mueren cuando los microorganismos mueren, no dejan un registro como los otros macroorganismos, es decir, no dejan fósiles en los sedimentos, no dejan esa huella. Cuando un microorganismo muere, todo se degrada, las proteínas, el DNA. Sin embargo, tiene en la membrana celular estos lípidos, lípidos que son esteroles, conocidos como algunos son el colesterol, como sabemos que tenemos en las membranas celulares, y este colesterol está formado por estos lípidos que son unos anillos de triterpenos, y estos triterpenos son muy difíciles de degradar y se quedan en el registro fósil por millones de años. Y estos marcadores de lípidos biomarcadores se quedan en el registro fósil, en el sedimento, y después se puede extraer estos lípidos para poder identificar qué tipo de moléculas eran, y después asociar con organismos que viven en la actualidad que pueden producir estas moléculas. Realizamos un estudio acerca de los lípidos que pudieron haber tenido estas Asgard Archaeas y encontramos diterpenoides, que son moléculas más pequeñitas, pero que hasta la fecha no se han buscado en el registro fósil, es decir, se han descartado completamente del registro fósil porque no se había encontrado en las arqueas estas moléculas. El único microorganismo que es capaz de producir estos diterpenoides es Mycobacterium tuberculosis, que lo utiliza como un mecanismo para no ser fagocitado por una célula del sistema inmune. Entonces, claro, mientras más buscamos y entendemos estos procesos, más podemos conectar con la vida en el pasado y lo que puede ocurrir. Estos biomarcadores también pueden ser utilizados para buscar registros de vida en otros planetas o estos biomarcadores. A nivel atmósfera, no podría decir que el metano es un indicador. Por ejemplo, hay metano que, dependiendo de su composición isotópica, se puede saber si es producido a nivel biológico o es producido en la corteza por procesos que no involucren procesos biológicos. Sin embargo, las Asgard Archaeas requieren o viven en conjunto con arqueas metanogénicas y sulfatorreductoras, las que se tienen en cultivo. Entonces, aunque produzcan estos, aunque haya, digamos, marcadores que permitan estar en la atmósfera, estos se degradan muy rápidamente y no se quedan en grandes cantidades como para tener un impacto en la atmósfera y que pueda ser utilizado como marcador en otros sitios o en exoplanetas. Y bueno, aquí en función de la pregunta que nos hicieron, le voy a preguntar al experto en sedimentos antiguos, Professor Galen. So there was a question about molecules that organisms can produce at the atmospheric level to be used as a signature to search for these biomolecules in other planets. But yeah.
The most obvious one is oxygen. So we first see traces of oxygen based on the geological record. We think we see traces of oxygen in the atmosphere starting about 2.4 billion years ago. and that surely is a sign that cyanobacteria are producing enough oxygen to overcome all the different reductants, minerals that reduce it into sulfate or other molecules, such that it can accumulate in the atmosphere. Presumably cyanobacteria evolved earlier than that. In fact, we think we see geochemical evidence of local accumulations of oxygen in the surface ocean, maybe as far back as 3 billion years ago. So yes, I would say oxygen is a good one. People look for oxygen in the atmospheres of exoplanets as a potential indicator of life.
But not specifically of Asgards.
Yeah, Asgards would be tough, but Archaea in general, one approach people take to look for the earliest evidence of Archaea is to look for isotopic signatures of methane.
Mhm.
So biogenic methane is typically very depleted in 13 carbon isotopes, very low ratios of 13 to 12. And although there are other processes that can generate those sorts of signatures, they're often the very low signatures are interpreted in terms of biogenic methane, and the oldest evidence for that is about 3.45 billion years ago, about the same as the oldest evidence for bacteria in the form of stromatolites.
So yeah, we would say that around that time, Earth was already populated by Archaea and bacteria.
Yes.
Yeah.
Yeah.
And then millions of years later, eukaryotes appeared on Earth, and that's what we are trying to understand. The process of eukaryogenesis and the origins of complex life.
Planets that helps out play with them.
Yeah.
It's a eukaryotic name.
The corollary of Fermi's paradox.
Sí, sobre perseguir carreras científicas, uno a veces lo ve bastante lejos, pero todas las ciencias al final se relacionan una con la otra. Yo estudié biología molecular y me la pasaba en el laboratorio y no, aunque estaba fascinada por entender el origen de la vida, el origen de la vida compleja y estudiar ecosistemas que eran la última evidencia de vida en la Tierra, me llevaron ahora a investigar el fondo del mar, las ventilas hidrotermales, black smokers, y todo eso tiene una relación con astrobiología. Entonces sí, no, si algo te fascina, tienes que luchar por seguir esos sueños. we found the waypoint?
Orbit
300.
bridge, are you happy about them another 10 degrees to starboard, please?
Yeah, that's good.
Thanks, Jess.
About 200 meters from the
Sorry?
Valerie, I see there's a comment about DNA 1.4 billion years ago. We should clarify that we're not able to recover DNA from that far back.
Yeah.
But we can make inferences
We
about...
can make inferences, evolutionary inferences about what could have happened. But yeah, we are not able to recover DNA. But fossils, though,
Yes, we can recover fossils, and by looking at the DNA of extant life, we can infer what, for
Exactly.
example, Lika, the last
Yeah.
eukaryotic common ancestor, might have looked like. I think the oldest recovered DNA is just a few million years old.
And the process of making this evolutionary inferences is really important for us because by using and understanding modern day archaea, modern day organisms, we can reconstruct their past. We can reconstruct how the ancestors of this archaea might have looked like. And that's why it's so important to have a better understanding of this archaea, and we're just at the beginning of finding more, and the more we find, the more we kind of discover, and there's so many unanswered questions. In the room there's a professor here, Bas Bum, that has been studying this Asgard Archaea. Bas, what can you tell us about, when you started studying these organisms and now everything that we've learned over the past years that they were first discovered, and what are the questions that remain intriguing that we still don't know, and that the field should move forward towards certain directions to kind of answer these questions?
Anything else?
a very long question.
I mean, so in the last few decades since the discovery of Archaea in the 1970s by Carl Woese, I think it was appreciated that eukaryotes probably arose from an archaeal partner as well as bacterial partner. So the discovery of Archaea in the 70s sort of clarified that. And then the question was which Archaea might be involved. And I had been thinking about what that might look like before it was discovered, and published thoughts about what we might imagine that archaeal cell was. And then this amazing thing happened in 2015 where Thijs Ettema and Anja Spang and colleagues, doing a similar thing that we're doing from cores taken from the bottom of the sea off the coast of Norway near the mid-Atlantic ridge, took sample, sequenced the DNA, and discovered that there were these new types of archaea, which they called Asgard Archaea. And so there was many years where we speculated and wondered what these cells might look like, because they're so important in the transition from simple cells to complex cells, eukaryotic cells. And it was only in 2020 where Imachi and Nobu published their first paper showing these beautiful cells living in partnerships or stealing and sharing with organisms in a microbial community in Asgard Archaea they identified off the coast of Japan. And so in some way, the kind of work we're doing, taking cores, looking for DNA, sequencing, trying to find new organisms, and then trying to culture them and look at them as exactly the path that led to the discovery so far. And yet we're just scraping the surface of the bottom of the sea, and there's much more to find out. So I think it's hard, but the kind of thing we're doing, I think, still promises to reveal lots of new things.
Yeah. Still a lot to discover. Thank you, boss. And now it's time to a change in the watch lead. And bueno, me despido. Soy Valerie de Anda y ahora con ustedes se queda Jacob and Laura. Y bueno, disfruten de este dive y de este live stream. Y gracias a todos por seguirnos escuchando. Gracias a todos los radioescuchas por estar pendientes. Y bueno, un abrazo muy grande. Yeah, that's what I'm putting here. So.
He said he wanted the top 5, but we should take 5 cents as well. Okay. Okay, so leave it. Sure. Yeah, 27.
And then probably be
Great. And we haven't collected any yet.
No, we're still heading towards the... Yep.
Hi everyone. I'm taking the relay here alongside with Jacob. My name is Laura. I'll be logging in. I'm just jumping in just to answer a question that I see in the chat about the origin of life. And I just want to clarify that what we are studying here is more the origin of complex life, and that those two events are actually very, very far apart in the history of life. There are probably a couple of billion years between those two things, between the very first emergence of life on Earth and then the evolution of complex life. So what we are trying to understand here probably doesn't tell us very much about the very, very first emergence of life on Earth.
We will before we sit down. We'll take 1 and 2. Try to find a patch with some open space. This looks pretty good. I mean, for what we have, it's pretty good. Mm-hmm. I think this is about as well as we can. According to the waypoint? Sure. Which would you prefer? I mean, do you want us to be like within a meter of the point, or do you want us to be looking for a place with, you know, fewer stars? Me personally, or
was a total
9?
Okay, so what we do is we can go wherever I can go in for like 3 or 4, and then move 3 meters to the right, do 3 or more if there's getting complex, so we don't get any stars.
Okay, yeah, it would be nice if we could keep them all. You know, I think it's kind of a priority to keep them close together.
Within how far?
A couple of meters.
Yeah, that's all
I know,
I'm saying.
you're not. Yeah.
Yeah, so we'll do here, here, here.
Sure.
Just to keep 2 view places.
2 batches. I think that's fine.
So this can before we land.
Please. 1 and 2, please. Buenas tardes. Han cambiado los pilotos y también los científicos en el cuarto de control. Me llamo Jacob. También hay Laura y Ian, Chris y Joel. Están pilotando. 1 and 2. Thank you. Niskins 1 and 2.
Niskin 1 and 2, yes.
Let's do port side first, please. No, port side. Yeah, port ram.
Okay, do you want all 6 on the porch then?
I'm just double checking that.
Or
I don't-
those are yours, and the other 2 I think are just normal.
I'm not sure we're deploying all 6 on the porch. I have here 1, 2, 3, 4, 5, 6, 7, 8, 11.
Okay.
So I think 9 and 10.
Yeah, 4 of them are yours and 2 of them are just normal, whatever.
Yeah,
So, alright, so I'll do all the ones on the port and then just the special ones on the porch.
that would. Yes,
Okay.
please.
I think they probably move away once you get close.
Maybe.
Sí, hay uruguayos en nuestra expedición, pero en el cuarto de control ahorita mismo solo tenemos Gaby, nuestra observadora del Ministerio del Ambiente. Pero ayer
RV Bridge, am I okay to come 10
tenemos
degrees
todo el
west?
equipo uruguayo aquí.
Yeah, that'd be fine.
Thank you.
Y es posible que ellos vemos más después hoy.
All right, mate.
So 1, 2, and 3, we want to go full length. Val emphasized this. Estamos haciendo una esfuerzo para evitar los sulfuros. Es poco difícil, pero es posible. Con precaución. Perfecto. PC-01 deployed.
PC-01.
So this is a really interesting question that's been brought up concerning oxygen. A lot of people kind of consider it elementary that oxygen is necessary for life, when in fact it is not, and life existed on the planet for a long time before there was any oxygen. In fact, it was created by life. So just some general background. To speak more directly to the question, though, inside of hydrothermal vents, yes, the environment is anoxic. You have these very oxygen-rich compounds, mostly sulfides, that will essentially react with and absorb any oxygen that's present. There is around the vents, as opposed to inside of them, you rapidly reach an oxygenated environment. Here, obviously there are not any vents, but we still have a really strong oxygen gradient, and that's simply because the oxygen can't travel very far into the sediment. It doesn't flow freely through the sediment, and there are a lot of microbes in the sediment consuming oxygen. And so interestingly, in some of these sites, including this site, When you go just a few centimeters deep, you end up with this environment that's very rich in sulfides, totally free of oxygen, and chemically, in some ways, very similar to a vent. We're interested in these places where oxygen-free and oxygenated habitats come up against each other because we think this might have had a role to play in the emergence of complex life. That's because, as you've heard many of us talking about, Asgard Archaea and their bacterial partners, including Laura just a few minutes ago, we think that the major role of the bacterial partner then, and still today, is to give the host cell a way to use oxygen to generate energy. So the bacterial partner became the mitochondria, which is where the majority of oxygen is used in the complex cells that make up you and I and other complex life. Push core 2 deployed.
Yeah, push core
Thank
2.
you. Ahead of me.
What is stated in the question is very correct. So most Asgard Archaea that have been discovered so far have been found in anoxic environments, and we do know a few of them that are have been found in the water column, and so here we're trying to find more of them from oxic environment.
Thanks for the direct answer, Laura.
Yeah, well, I mean, what you said is context, is the right
I tried to
context
set
for
the
why
stage, but I didn't really answer the question.
we're doing this.
Yeah. PC03 deployed.
PC03.
You know, it's very, very small stuff. So I wanted to, I'm sure how well it's going to come out of there.
Okay. Well, we have spares.
Is it worth trying one?
And these a little bit of a gap.
What's that?
These leave a little bit of a water gap.
Yeah. So those are the 3 full lengths, and then this one we want to leave about a 5 centimeter gap. Ok, puedo tratar de explicar este fenómeno de vida aeróbica y anaeróbica. Los ambientes hidrotermales, sí, en los... ¿cómo se dice? No recuerdo.
Nice driving.
En los vents no hay oxígeno, pero hay una frontera entre ambientes anóxicos y óxicos. También en esta locación hay una frontera entre aguas oxigenadas y el sedimento anóxico. Estamos interesadas en esta frontera porque los Asgard Archaeos que ya conocemos todos son de ambientes anóxicos. Y buscamos para nuevos arqueos, Asgard Archaeos, que son óxicos o parte óxicos. Y nuestra interés es porque creemos que el evento del comienzo de la vida compleja tiene algo es relevante metabolism de maneras metabólicas óxicas. La bacteria en la interacción ancestral entre las bacterias y los Asgard Archaeos, es probable que momento. Tenemos un ángulo.
Se cree
Sí.
que la bacteria en el Archaea
Sí.
se
Creemos que la bacteria ayuda al arqueo para usar oxígeno para generar energía.
I'm going to collect these and then do the torch afterwards.
I think so, especially because of that angle. Yeah. Yeah. Let's get a shot of these deployed, please. Sí, no tengo mate o café suficiente.
y por eso se cree que las arqueas Asgard existen en environmentos oxigenados también. Y sí, es porque los buscamos en están environmentos.
Estamos unos kilómetros hacia la plataforma continental de Uruguay. Estamos más o menos en el pared de la plataforma. Y sí, los Asgard Archaeos ya tienen sus compañeros bacteriales. Las culturas que ya existen en laboratorios son coculturas con arqueos y también bacterias. Y la teoría prevalente es que la junta del Asgard Archaeo y bacteria comienza la vida compleja. Push core 1 recovered.
Push core 1 logged. Yes, so I will first answer that question in English and then we will poorly try to translate. So there's a question about how oxygen and bacteria are crucial for the development of complex life. So this is still something that is being that we are still researching. But yes, it's the main theory right now. So if you look at complex cells, one of their characteristics is they all have the way that they make energy is that they have inside our cells, we have mitochondria, and it's a small compartment inside the cell that makes energy. And this compartment was originally a bacterium that was engulfed and sort of tamed to now it's not a bacterium anymore because it's not independent, it's not self-sufficient anymore to live on its own. It's been really sort of tamed by our cells, but originally it was a bacterium, and this is really why we think this was the first event or a very important event for the origin of complex life. And the way that this compartment makes energy generally is by using oxygen. So this is why those two elements are very important. I hope that answers the question. Sorry, I will translate in 1 second. Trying to do two things at once. En español. La pregunta de la importancia del oxígeno por el origen de la vida compleja es que si en las células complejas
Perdón, Laura tiene el trabajo más importante en este cuarto después de los pilotos. Ella necesita recordar nuestras muestras.
Tenemos mitocondria, no sé cómo se dice en español, mitocondrias, que todas las células complejas tienen mitocondrias y que hacen energía y por hacer energía usan oxígeno y mitocondrias originan de bacterias. 2 millones de años mitocondrias eran fueran no sé.
Se echan las mitocondrias. Sí. Sí. Hace 2 millones de años fue el origen de las mitocondrias, ¿sí?
Y mitocondrias origen de una bacteria.
have 2 more regular sleeves on the porch?
Yeah.
Okay. Let's
All 6.
yeah, let's do those, and let's try to do them with about 5 centimeters at the top.
Okay,
I mean, you
so-
notice the sediment's really soft, so you have to go
Yeah,
with
so it's compressing. So
Yeah,
the more I
exactly.
push down, the more it will compress. So it's not gonna make it's not gonna, the reason that the last one was higher was because the angle that I went in, so it didn't compress as much. Because it is so soft, it will compress as I go down.
Yeah.
So even if I go all the way down, it's still gonna be
Sure.
significant amount.
Yeah.
Alright.
Given that we know that, I'd say just go all the way in. Thanks.
Guys want to move a little bit forward or somewhere else? Like the rest. Is it in your head or?
Let's. How? I mean, I'm looking at this patch over here. Would you have to pick up to access that? Can you reach out here, or would you have to pick up and get there? You can get there? Yeah, why don't we just do that? Before we make a ton more of a mess, could you please, do we have any cameras up near the Niskins? Oh, I see a lot of schmutz coming through our starboard thruster.
Yeah.
Yeah, so there's a lot of stuff up in the water. Okay. I'm just thinking about how much mud we'll get in the Niskins, because Danilo came and asked if we could fire a couple more.
Okay, 2 more.
Why not? Yes. Thanks. Niskins 3-4.
Niskin 3-4.
You're gonna do the regulars or the specials first?
Whatever you prefer. You have a preference?
No.
Okay.
Ya tenemos 6 más cores. Nosotros ni tenemos no tenemos ni excusa para no comprender a ustedes porque tenemos una traducción de inteligencia artificial, pero no es bidireccional. Además de esos peces intricadas. Creo que son juveniles. Somebody did a terrible tape job on that one. It was me.
So no, most
Sorry?
it's pretty. Someone is
No, just Ben Irwin.
saying that they thought most were Spanish speaking.
That's perfect.
It is very random. We do have quite a Uruguayan crew among the scientists here, but it's pretty international. We have people from Czech Republic. We have many people from the U.S. I'm from France originally, and so our usually in the scientific community the shared language is English. So some people speak Spanish, some people don't, but it's pretty random. So depending on who you get in the mission control, some of us do better than others at speaking Spanish.
I'm trying to find places that don't have seafloor, right? That's good right there.
Nice job avoiding the stars. I'm impressed. Mm-hmm. You can go right up to the end of the tape, I think.
What's that?
Right up to the end of the tape, probably good. Like another centimeter. That's well, thank you. Beautiful, those juvenile rat tails. I sometimes wonder how much ground a rat tail covers in an environment like this. The little ones just go with the current. Todos estos cores tienen sensores ópticos de oxígeno. Es abajo de la cinta negra.
Are you okay with me bringing the heading around 10 degrees to 4?
Yeah, but it's no problem. Go ahead.
Thank you.
That's good, thanks, Chris. So this one, try to leave us some headspace, these two. More headspace, I think, than you left me. Yeah, tagging rat tails would be pretty awesome, but you run into that radio waves underwater problem. Not to say that it's impossible. Hopefully someone figures it out someday. Okay, that's good for one. I have specific requests for two that have some space in them, and from last time, you know, the like one went into a slope, so rather than have somebody be unhappy with it, I figured we'd just take backups. Las puntas blancas son conchitas de un forma a otro. No recuerdo precisamente de nuestra visita previa a este sitio, y fue en un lugar unos kilómetros de aquí. Pero creo que son conchitas de gusanitos, o es posible de molusquitos. Vamos a ver cuando recogemos las muestras. Hay un tenóforo en la media distancia. Es una especie poco atípica para este ambiente, del género Bioluminescence. El tenóforo es ahogado, pero puedes reversar su video. Okay. No, that's perfect. I was just going to mention to Laura another instance instance of channels in the sand on the deployment of number 6.
Yeah.
You can just put it in the notes for that.
I will. Yeah.
Thank you.
Sounds good.
Idealmente, las muestras, actually, that question was in English. Ideally, we get the samples pretty close together because we're all doing different sorts of analyses on material that we want to be similar, so that we can cross-reference our results with each other. You know, and for instance, see if, so we could link, for instance, the DNA of certain microbes to the membrane molecules they're producing, things like this. In practice, we don't want to disturb the brittle stars, so we bank on the sediment being pretty consistent within a few meters radius, and we put the cores where we can. You'll notice there appear to be some open channels under the sediment, because sometimes when we push in these cores, you see some dust popping up at a location a few tens of centimeters away from the core. I think that's good. Thank you. The question about oxygen and a huge jump in available energy, I think, is basically spot on the prevailing theory. Yes, we do. Thank you. Can we get
Yeah.
all push cores deployed, please?
Yep.
Call out when you're ready.
Just 1 second.
Yeah.
You happy?
No rush. Not quite.
Oh, good. Thank you.
Okay, we're happy. Thank you. So yes, it's thought that the ability to use oxygen for respiration allowed for sort of a huge evolutionary breakthrough in the complexity of life. However, you ask an interesting question about anaerobic complex life. There are eukaryotes that can make do with various amounts of oxygen. Some can make do with very little, and some of the complex life that lives in the water column, similar enough to the ctenophore we just saw drifting by, they can deal with surprisingly low amounts of oxygen, and some of them, in fact, cannot tolerate high amounts of oxygen. But most of those animals, multicellular complex life, still fundamentally use aerobic respiration, which is the type of metabolism that came out of the Asgard bacterial partnership. Tenemos una pregunta sobre si la moda metabólica aeróbica hace posible la vida compleja, y sí, eso es la teoría prevalente, que después de la cuenta del Asgard Archaea y la bacteria y el origen de las mitocondrias, sea posible posible tener la vida compleja y animales y todo eso. That was... Guessing you got 9 recovered?
Yep.
Thank you. Very, very soft. Personalmente, mi animal, mi tipo de animal favorito es son los dinóforos. Toda mi tesis de posgrado fue sobre los tenóforos y sus adaptaciones a la alta presión. You have a favorite animal, Laura? Favorite protist? Even better.
No, I love them all.
Todos los animales son los niños de los arqueos y protistas. Are 9 and 10 seated?
I'll probably look at the bottom, but I'll put them down as soon as I get done.
Okay, 6 just looks a lot snugger. PC7 recovered.
PC7.
I think I'm less than 50% on calling out samples to you.
That's
So
fine.
Thanks for
I think
staying on top of it.
so far we're okay.
If they're not definite, we don't trust more.
Sorry, could you repeat?
I said, definitely not gonna go any deeper.
Hmm. Why? Don't think
Push
it'll.
on it and move it.
Oh.
I mean, it could be that it's always heated, it could be that it's hot outside the floor over. I'll look at it with the arm almost,
Sure.
just to see if I can get a.
Yeah, it looks a good sight higher. I don't know if you think it's caught on the lip of the stopper or
Yeah.
the sediment wasn't falling out.
Bring me
Here.
Tenemos una cámara en el brazo y con esta estamos chequeando la locación de dos de nuestros cores, la posición.
I'll try them again in a minute.
Okay. Two beautiful layers in there.
That's so much for
Hmm?
this one.
Hmm. The focus
Not so
on its own? Yeah. What elements is in what kinds of chemicals? What sorts of food do they eat? Just want to make sure I answer the right question. I think most of them eat. Well, this is not going to sound very specific, but they eat organic molecules, just like you and me, sugars, proteins, etc. And there's plenty of that for microbes in all of this material falling from the surface. Laura might be able to give a more specific and interesting answer.
It varies a lot. So those microorganisms, I mean, when we talk about them, when we are right now talking about them, we do tend to generalize a lot, but it's a very, very diverse, it's a huge group of organisms, so it really depends on the subgroup that we are looking at. We are still learning a lot about them. Most of them we've never managed to grow them in a laboratory conditions, so we don't necessarily know for sure what they eat, but from what we can guess is what Jacob just said. For most of them, they eat small organic molecules, so small proteins or small sugars, that kind of molecules. But it really depends on which groups we're looking at.
Beautiful work, guys. Those look great.
This was 08, was it?
That was 08.
Thank you.
Is there any kind of like, or is that more liable to mess it up if we wiggle it around?
Just shake the.
Mm. Oh, there we go. Okay, there's 1. I'm not convinced about 9. Okay. Los pilotos no tienen nada información táctil del brazo. Tienen que operar solo visualmente. Y por eso algunas veces requiere
That's just what it is.
unos tratos. Sure. No, I mean, I think it's a good batch. We got a couple backups, so we're happy. Thank you. El verdadero Carlos en este buque es nuestro sous chef. Hace comida deliciosa. Okay. I think we're all set for samples. Did you get any information about off bottom time? Because I didn't.
It said on deck 5.
Okay.
I think. I can go verify that.
I can check that on our daily plan. I've just been moving around a lot. Yeah, I have 1700. How long is it going to take us to come up?
Where are we at? 900?
I would say the city an hour.
An hour?
Yeah,
So off bottom in an hour from now. It's almost 1500. Yeah.
Yeah, so between 35 and 40 minutes to get the local all the way up, and then add another 20
Sure.
20 to 30 minutes depending on whether we get us back on deck. So yeah, I would say we probably would be off about 4 o'clock until right.
Great. Okay. We can give you longer if you want to, but that means we have about an hour to explore.
So, check Mike and see if he's good for that as well.
Sure. There's been some interesting stuff drifting along the bottom here. Hola, bienvenidos. Estamos poco afuera del plataforma continental de Uruguay, en una profundidad de más o menos 900 metros. Tenemos todas nuestras muestras para esta inmersión y vamos a explorar para una hora más o menos. Unas de las vistas prominentes de esta inmersión son los grenadiers, los rat tails. Yo lo siento, pero yo olvido el nombre para estos peces en español. Pero tenemos algunos que son juveniles y tienen rayas muy largas. Is the Zoom still acting up? Is the Zoom still acting up? Or is it okay? It's okay? Let's check out these red things on the bottom. We saw one before. You almost zoomed on it. That right there. The red snotty thing. You saw one earlier. Peces ratas juveniles que parecen más intricadas que los adultos. Yeah, let's zoom on that. I don't. Oh, it's a dead prawn. You're right. Well,
Now we know.
el ciclo de la vida. Okay, that's all right. Rico con moléculas orgánicos para los arqueos y bacterias. Yeah, we don't have to keep staring at the dead prawn, but no. Yeah. This is biogeochemistry in action.
Yeah.
It looks like there's a live crustacean up there, but we can keep chugging.
Can go to the peces rata. It's now you know.
Yeah. Someone told me a different name yesterday too, so now I have granaderos, peces rata. There's another. Granaderos makes sense, and another English name is grenadier. And there's your French version for these fishes.
Yeah, I don't know.
Well, no, I think it's probably grenadier, I would guess.
I think so.
But,
Yeah.
yeah.
Grenadier, yeah.
Grenadier. Are we stopped on something, or are you just getting set up? Is it still? Looking at this worm.
There's an Instagram. Someone is asking about Instagram for this expedition. It's just the Schmidt Ocean Instagram, but they do post pictures from the dives.
Can you mark this as a worm, please?
Yes, sorry.
No, you're fine.
Yeah.
We're here, so.
Tenemos un Instagram, el del Schmidt Ocean Institute.
We can zoom out. También con el zoom podemos ver unas conchitas de moluscos en la arenita. Hard to say. Could be a ribbon worm, an emerdian, without disturbing it. It's a little bit hard for me to tell. Certainly didn't look like a polychaete. Sí, estos son un tipo de estrella del mar que se llaman ofiuros. Hay estrellas, pero son en una clase diferente que la mayoría de las estrellas que viven en la playa, por ejemplo. Todos los ofiuros son más delgados de las otras estrellas más rápidas. Y también se pueden desconectar sus brazos como una moda de defensa. This is some type of soft coral.
Okay.
I don't know exactly what type. You can mark it. Oh, it's a sea pen with like a weird shape.
Yeah.
Okay, so you mark it as an idarian,
Yeah.
but not properly a coral. Huh. I haven't seen this one.
It's pretty.
Oh, there's another one of those benthic ctenophores.
Yeah.
If you tilt down a little bit, un ctenóforo bentico. Hay muchos y muchos en este sitio. Estamos un poco afuera de la plataforma continental del Uruguay, en una profundidad de casi 900 metros. Estamos explorando al fondo y ahorita mismo tenemos una pluma del mar, un tipo de nidario, y también una especie de ctenóforo bentico. Y los ctenóforos y los nidarios, los dos tipos de animales aquí que no son ofiuros, que no son las estrellas, los dos son gelatinos, pero no la relación evolutiva entre los dos es muy, muy distante. Es poco como una coincidencia que los dos tienen cuerpos gelatinosos. Oh, we can look at it. I mean, I'll stare at benthic ctenophores all day, but nobody else wants to do that.
You waypoint, thanks, Chris.
Hello, Captain.
Hi there.
Yeah. Go for it.
There's another type of star.
That's good.
Starfish. Oh, yeah.
Oh, there's a squid in the left. All right, take a look at him. Everybody loves a cephalopod.
Oh.
Nice. Just hanging out.
Alright, Joel, scoot over down this route.
Doing the same thing.
Down
Let's
this
see what it looks like.
Interesting squid behavior.
There's some that will just sit like that. Seems like we've been getting decent amount in the South Atlantic.
We're lucky to have Alex, our resident squid expert, in the control room at the moment. I kind of think you've earned that title.
I've seen a lot now.
More than most of us can say.
I do like them.
Después miramos este calamar. Podemos usar los lasers, que son puntos de luz con 10 centímetros de distancia entre. Y podemos ver el magnitud de las estrellas.
Maybe.
Un momento para claridad. It's like patience pays off. Look at those. Are those superorbital photophores, or is it? I think it's iridescence that we're seeing.
Mm.
What do you think? You see the eyebrows?
Yeah. That's here it does. Like all the little chromatophore patches.
Yeah. Con nuestro zoom podemos ver los cromatoforos, los células en la piel de los calamares que pueden cambiar su color. Y también los iridoforos, que son células que pueden cambiar la propiedad de reflexión en el piel del calamar.
Bye. Well, still there.
Okay.
Thanks for the camera work, Oil.
Yeah.
We have some annotations on this guy, yeah? Or gal, can't tell.
Can we have the lasers?
I had them on earlier.
Oh,
See 'em, but
sorry.
Yeah. Oh, there they are. Okay, so podemos ver los lasers en la cámara de los pilotos. Y por eso este calamar es más o menos 20
yeah.
centímetros.
Oh yeah. Okay, it's not that big.
You have any idea what this behavior is about, Alex?
No, we see them just sitting on the bottom like this sometimes.
What's that? Movement.
That's breathing.
Breathing?
Yeah. Yeah, they use the same muscles for breathing and propulsion. You know, on the bottom, like under the tentacles, there's a little funnel, a little siphon, and that's where water comes out when they breathe. But if they want to jet, right on cue.
He's like, no, I'm out of here.
Look at him
standing like that. That's cool.
Yeah. It's like it reminds me of cuttlefish.
Yeah. Oh, like the flambeaus that'll walk along. And stuff.
Yeah. I mean, there are a lot of sepioids that do that.
Yeah.
It's
I
maybe
I enjoy them.
the missing link.
Creeping along.
You saw commercial about
I know.
an outboard engine, small
It's not
engine
good.
that is using. I saw commercial of it. They're using this same principle as the jellyfish use
Mhm.
propulsion, like a diaphragm
Mhm.
expanding of it. Creates
A jet.
stream,
Yeah.
stream. I don't know how effective it is.
I think Kakani Katija is going to be out on a cruise with you all soon, and she did some of the original papers on the jetting dynamics of jellies. could ask her about that engine. She might know about it. No estamos 900 kilómetros hacia la costa. Tenemos 900 metros de profundidad, pero solo estamos unos cientos de kilómetros hacia la costa.
Can we continue?
Yeah, we can continue. This squid's about 20 centimeters in length. We just lasered it a little while ago. Yep. The lasers here are 10 centimeters apart. Of course, this will only tell you how wide the squid is, but you get the idea. You said you saw something else cool. It's probably gone now. Squid always stalking the show. Sure.
Jacob, can I ask a quick question? I was gonna, have you guys fired any Muskins?
We fired 4.
Great. Okay, cool. I was gonna get set up.
2 before and 2 after.
Lovely. Sounds good.
Yeah.
Cool. I just wanted to see a connection. Go ahead and get started. Thanks.
Sure. Any idea what kind of squid that was? You have seen a bunch of them this cruise?
We saw more last cruise, I think.
This is skip camera. Right. Skip camera is not coming on.
Yeah.
Oh. So we leave it off now.
That one in German.
That camera is on.
Are you trying to stay outside the donut?
Yes, we have.
Okay, yeah. Oh, it's one of those. Okay, this one, it's a fish. I think it's a king of the salmon, which is the most bizarre common fish name I've ever heard. You're like, that's not such a weird.
Yeah. You saw one on the way down.
Actually, that's not. No, I think it's a juvenile rat tail.
Yeah.
Yeah. We saw a few more.
Well, we saw one right before we touched bottom, and then there was just a million rat tails everywhere. So. We're gonna fight that focus. There we go.
Nice. Fight the focus almost as much as he's fighting the prop wash. Beautiful photography.
Yeah. It's too
Yeah.
close to us now. It's not too...
Looks like he's doing okay for himself. So this is, and thanks to another of our listeners for the help, this is probably a juvenile macrurid fish, similar to all of the rat tails that we've been seeing. Es un juvenil de los grenaderos, el tipo de pez que hemos visto toda esta inmersión.
Okay. You want to go?
Never mind.
I imagine there's a few of them around.
No es sifonóforo, es un pez, pero la forma de ese pez es poco similar que un sifonóforo. Es un ejemplo de la convergencia evolutiva. Sí, un pez, un pariente acerca de esta especie.
Yeah, that squid could have just been a short fin squid.
Mm.
Argentine short fin.
You know what? In the camera, it's actually good. It's full motion.
Actually, no, I saw this on earlier. You're right. Yeah. I wonder if that's what it was. They were saying it wasn't working earlier because I feel like the edge of it.
Yeah.
I thought it just wasn't the light that was on it, but get it.
Good to know.
So, pick something up?
Thanks for coming.
We'll be very specific about it.
Ya tenemos más o menos 40 minutos para explorar.
Una big waypoint bridge.
Ahora en el cuarto de control tenemos Alex, Chris, Joel con el vehículo y Laura y yo, Jacob. Son los
Very Charlie.
científicos en el cuarto de control para estas
Put up a
2
waypoint
horas.
for you.
Copy, thanks, Chris.
So does the ship usually drift when you're flying? Does it, like, do we not go into DP when we dive?
They'll be in DP, but we'll just move them as we go, so they'll stay in
Let's
DP
do
and
it.
then just set it up with that.
Yeah, so like there's the little purple, or over there it's yellow, but the FKT.
Uh-huh.
The pilots can move that, and then the bridge sees that waypoint.
And changes their waypoint.
Yeah.
Yeah,
Cool.
so we line up the launch recovery wherever we want right there and keep that the distance that we've flown from the ROV.
I've just heard a lot of talk about drift of the ship. Maybe I misunderstood
We've
it.
been doing drifts for our launch recoveries.
Mm.
We need to be able to maneuver if the current's too strong.
Right.
We can't overcome it, so we bring the ship in line with the currents, and it's easier for us to get away from it or be to it.
That makes sense. Less relative current.
Yeah. And then when we're on descent, we don't really care that much about location. So we'll have to drift in that time.
Makes sense.
So I just go into this.
We can. Yeah, we can look at it. Tenemos otra anémona y si tenemos buena suerte es posible que haya más tenóforos en el fondo. Sobre. Pero parece como una anémona común. We can annotate as an idarian.
Yep.
Hey, Brett.
Hello.
We got all our cores. We got a couple backups.
Cool.
We're just twiddling around until about 4. Does that sound reasonable?
Sure.
Off bottom?
Yeah.
The pilots are telling me.
Sure.
Call out when you get your shot. Oh, there is a Tina 4 behind it. I called it. Bonus points to anyone who could spot the Tina 4.
It's right there on the top.
Yep. 12 o'clock.
Yeah.
Brett gets a bonus point.
Whenever you get the shot, we can.
Yeah, I got
okay,
it.
great.
Yeah.
Thank you.
The left side is very pretty. The rest of it looks kind of weird.
We can go.
Wait, is this thing?
Hello and welcome to any listeners who have joined recently. We're on the continental slope off of Uruguay at a depth of about 900 meters. We're here looking for Asgard Archaea and their symbiotic partners. These are the sorts of organisms that gave rise to complex life over 2 billion years ago. But since we've gotten our sediment, in which we're going to search for these microorganisms, we have a little bit of bonus dive time to look around at the local fauna. Muy buena pregunta sobre los nidarios y los tenóforos. Y la respuesta es que no son parientes. Parecen similares, pero esta es la convergencia. La distancia entre... I have to think about this. La distancia evolutiva entre los nidarios y los tenóforos es misma que la distancia entre nuestros y tenóforos. Y estamos parientes más acercas de nidarios que de tenóforos. What's that? It's maybe another pen or something. Yeah. Y creemos que los tenóforos divergían de todos los otros animales hace unos 600 millones de años. Más o menos 50 millones de años. Sí, estamos más comunitarios que tenóforos. Parece extraño, pero es verdad. Las cosas blancas grandes son ofiuros, que son un tipo de estrella del mar. Pero también hay puntas blancas y creo que estas son conchitas de algún tipo de animal. Es posible gusanitos o mosquitos. think that our last scheduled diving day is the 2nd. Is that correct?
I got
Max would know.
a message this morning that might call that one. Let me see.
Sure.
Uh, 2 a.m. on December 2nd, we need to break off from the farthest station. In theory, we'd be in the closer station, so I guess it depends on where we are. If we're further spot out, then we wouldn't have to
Mhm.
on the 2nd. If we're closer to the house.
Mm-hmm. Yeah, so we plan to have 1 or 2 more dives on this expedition. It's going to depend on the weather and where we're able to dive. Thanks for the lowdown from the pilot's end, Chris. So we have 2 more on our wish list, but let's see what we can manage.
We're going to month of the day of Buenos Aires, so there's a lot more flexibility.
Same, you can, just doctor we went to Buenos Aires. Same
We're going to Montevideo, so.
Yeah, but
With the alien. I think we want to say it's
Oh, stop, please. See this? I think it's a... Oh, yeah, it's a... Es un kit... ok, va a ser femenina. Es un ketognata. O es posible ketognato, no sé. El nombre en inglés es un...
I think it's the same, so I think you're right.
Es un animal muy acerca que parece como vidrio. Yeah, very hard to see. It's up on your dome. Yeah. Do you know? Anyone in here know how to say arrow, Sissy?
flecha.
Fletch? Yeah, yeah. Oh, no, Sissy's back here. Okay. Sí, en inglés se llama un gusano flecha, porque parece poco como... Sí, de hecho. Por la mayoría, los ketognatos, los gusanos de flecha, son pelágicos, pero hay algunas especies bénticos. Y todos son predatorios. What's that? Oh, sorry. Do you think it's more dead shrimp? Or a biomat? Oof. We might actually be interested in that. El ketognato se va, pero es probable que va a ser mucho más en la subida. Pregunta interesante sobre la cantidad de ofiuros. Por la mayoría, los ofiuros son detritívoros, comen el detritus. Aquí es obvio que hay mucho detrito.
Wow, that's a lot of stars.
Y hay una parte del sedimento que es que contiene muchos químicos orgánicos, es como esponja. Y creo que eso es esa cantidad de detrito, de de comida para los sulfuros es parte de la razón por qué hay tantos. También hay mucha de la del nieve marino en los otros sitios más profundos, pero es probable que el nieve en esos profundos extremos es peor como un tipo de comida. There's another ctenophore. Yeah, look at this guy. Fish, the fish. Yeah. I want to look at this eukaryote. Yeah. Sorry, I only get I only get sassy when there's a big ctenophore.
I was pointing at it, you just said you were pointing at it.
If we could get some close-ups of this, I'd appreciate it. Please. I mean, yeah, I guess I'm asking both you and Laura.
Yeah.
It's not an easy close-up to get.
I did get one.
Yeah,
but
great. Hay unas especies de ctenóforo que son bentopelágicos, que significa que solo viven acerca del fondo. No estoy cierto si eso es uno de estos especies especializados. Pero cuando buscamos estas especies bentopelágicos, la estrategia para verlos es para buscar la umbra en el fondo. Sí, la mayoría. Oh, yeah. If you could snap a close up with the frame grab, I'd appreciate it. But
Yeah.
then we'll let this guy in his way. This one has eggs. So those little pouches alongside the comb rows. Casi todos los ctenóforos tienen bioluminescencia. No todos, pero casi todos. Pero lo que vemos ahorita mismo es iridescencia. Es una fenómena similar que lo que pasa con aceite arriba de agua. Se hace por interferencia entre rayos de luz. Los ctenóforos tienen estructuras que son como los dientes de un pene, y esto es el origen del nombre ctenóforo, que significa pene. Cteno significa pene. Estas estructuras como dientes de pene son no sé, son muy finas y por eso producen la iridescencia. Sí, muy similar que las plumas de picaflores. Are we Are we? Hold early. Huh? Oh, okay. No, I just saw something about weather.
No, just a link for it.
Oh, to what the weather is. Nice. Actually, the exclamation point got me excited.
Feels remarkably better than it did in water.
Good.
For Joel's sake,
No.
Only have one tail something.
La manera en que le describí es poco confuso, lo siento. En esta especie no son los dientes del animal, solo tiene un nombre con origen en los dientes de un pene, pero es de verdad son más como, how do you say fin on a fish? Más como aletitas y se usan para nadar. Hay unos especies especializados de tenóforo que usan unas de estas estructuras como dientes para comer, pero no la especie que hemos visto. It's probably just another C pen. I'd like to get a look at one of these big red masses next time we see one. The ones that don't look like animals. Like there's one, we're over it now, but okay. I'll point it out when I see another. I think that is. I can't see it from above. It's above the fish.
No, I saw these arrowfish.
Oh, yeah. Yep. Yeah, I saw it too. They're so hard to focus on. I've sectioned them before, but the species that we were catching, they were orange, which makes it much easier. We're just getting a look at this bio mat here. Sorry, are you about to say something, Alex? Mm-hmm. Oh, okay.
No, I'll say something.
Nothing.
Yeah, that thing in the foreground. There are these sort of red amorphous masses that I've been seeing on the bottom, but we haven't gotten close enough to tell what they are. Well, whatever this is, it must be tasty because the brittle stars are all over it, and you can also see a bunch of snails eating it. They're little brown things. I don't think we can take it because there are too many animals tangled up in it.
We sucked some of that up earlier this cruise.
We did. Well, wait, who knows if it was the same thing?
Yeah, we suctioned goop.
Yeah.
It's more like July.
It's very stringy.
Yeah, we really need a dichotomous key for goo, schmutz, muck, and snot.
Yeah. I don't think that's microbial though. I think
No.
it's more animal made.
Yeah. Ya. Hay unas posibilidades sobre que este puede ser una casa vieja de un larvacio. También sería una medusa muerta. No sé, hay muchas posibilidades. Pero no es un animal vivo ya. Y el movimiento es porque del growth. We can move on. I don't think we're gonna learn anything else about it. I think it's most like.
I don't know what it'll be like once I got it.
Yeah, let's keep looking. I've seen some other interesting mats. We don't like Brett is interested in them because they're microbial, hotbeds. But we've just frozen them, the sequence for DNA, so it's not like there's a imminent need.
Cool. What are your thoughts about once you get clear that I'll show you.
Oh. El animal con la parte roja es actualmente un camarón. Y es todo transparente excepto por el estómago y otras partes que son rojos. La razón creemos que el estómago es rojo es porque debe que ser opaco, sí, opaco para bloquear la luz de los artículos de comida, otros animales que son bioluminescentes. No, ya subimos, tenemos unos 10 o 15 minutos más. Es un pariente de krill, es un tipo de camarón. Another sea pen. Yes, those are the things. Thanks for spotting it. Yeah, I would love to figure out what that is. The last time we looked, it was a dead shrimp, but this looks too big to be a dead shrimp. Maybe it's two dead shrimp. Called it.
It's still shrimp. It might just be the molds, honestly. But.
Oh yeah, that's very likely.
Yeah.
Okay, now we know.
Yeah. That's cool though.
Mm-hmm.
Now, how do we say mold in Spanish?
La concha vacía de un camarón.
Muda.
We'll see. Nuda?
Muda.
Muda? You looked it up, or did somebody
Yeah,
tell us?
no, I did.
Okay. Gracias a Laura. Ya conocemos que esos son las mudas de los camarones pénticos. Son como pieles vacíos o conchas vacías.
How Updated waypoint bridge.
Okay, thanks, Chris.
La parte roja entre los camarones no es bioluminescente, solo es un pigmento que es opaco, pero refleja mucha luz roja. Naturalmente, en el mar profundo no hay nada luz roja. Pero en nuestro ROV tenemos luces blancos que tienen todos los colores, incluso rojo. Y por eso podemos ver los camarones rojos. Sin nuestras luces son invisibles. Okay, we boldly explored our unknown shrimp mold. En una de nuestras inmersiones previas, there's something right here. Can we look at it? It might just be a snot ball, but it might be cool. Are you able to lock on? Gonna feel really silly if it's a snot ball. Oh no, it's like, there's no way. It's, huh? Yeah, we lost it. It's okay. That was a Hail Mary. Sometimes you get these jellies and ctenophores that just float like centimeters above the bottom. You can only tell them by their shadow. En una de nuestras inmersiones previas en este sitio hemos visto unos elasmoblancos, unos tiburones y también unas rayas, pero no creo que hemos visto nada de estos hoy. No olvidan que si no hay sol en estas profundidades, no hay luz. Hay una cantidad sorprendente de luz, pero toda esta es bioluminescente. Más o menos 70% de las especies, en el columno de agua profundo tiene la bioluminescencia, y más o menos media 50% de las especies bénticos del mar profundo tienen su propia luz. What's the big dark spot out here?
It's
Just a fish? Oh, so disappointing. Oh, so it is. Yep.
Yeah, I was just as excited as you were.
Thank you.
Is it the Oreo fish we saw?
Mm-hmm.
they're cute, but
Yeah.
the one earlier was mean bugging for us.
I didn't know this one.
Nah, it's okay. Look at him just hanging out, minding his own business. Being 200 years old.
About 200 times as
Una explicación. Oh, para mudar el piel. Pues es una pregunta interesante. El piel de los crustáceos no momento. Tengo que tengo que considerar eso. Oh. Una explicación para bioluminescencia. Es un poco más fácil. Hay unas razones por qué creemos que los animales usan la bioluminescencia. This is speaking of. Habla de bioluminescencia, es un tipo de medusa bioluminescente del género Eukaryotes. La medusa de cristal. Hay muchos en el columna, pues. Podemos continuar. Okay. Gracias. Oh, more benthic ctenophores. So many. It's okay. No, just keep going. Yeah. Keep going in a few minutes. Unos animales usan la bioluminescencia para cazar su comida. Es como un no sé, cuando se pesca con como
Carnada por atrás. Carnada.
Es poco algunos animales, gracias a Joel, pueden usar gusano de flecha. Pueden usar la bioluminescencia como carnado para pescar. Otros pueden usarla como una alarma cuando un otro animal le ataca. Hay un sitio de web bueno sobre bioluminescencia que tiene un catálogo de estas adaptaciones bioluminescentes y la dirección de este sitio es Biolum, BIOLUM, .eemb.ucsb.edu. I'll try to pull a frame grab on this red jelly. George will know much better than I will. Probably has a frame grab already.
Did you just talk about hiding from predators? Is that what you just said? Because I was going to talk about that.
I was actually talking about. So I was talking. Somebody asked about like why animals have bioluminescence,
right.
Because it kind of seems counterintuitive,
right.
and maladaptive.
Right.
There's what some people call the burglar alarm model,
right.
which is that if you can attract a predator of your predator.
Oh, okay.
It's a little bit different than hiding. So no, go for it.
Una otra razón para crear tu propia luz es porque puede ser visto desde abajo porque proyectas una sombra y así que si proyectas tu propia luz puedes esconderte mejor de los depredadores. So when you are a fish or something else in the water and there's light coming from above you, you project a shadow, so you are very visible from below, and so you are visible to predators. So if you create your own light, you're less visible from predators, so it's a way to hide better.
Sí, un nombre para esta es contrailuminación, which you probably already said.
No.
No.
Thank you.
Es un nombre técnico.
This guy.
This guy over on the right.
Gracias, Laura.
You want to see the snaky?
Sneaky snake.
Did we take any water samples?
We did. We took 4, and we're happy with that.
Saw one of these guys on like the very first dive of the expedition.
So we have a deck time on the science end of 1700, which I think means we should come off bottom soon.
Yeah.
Agreed. Egregious. This is egregious. But now, just see.
Yeah. Shiny.
Sandy.
Muchas de las consecuencias de la evolución son contraintuitivas a nuestros humanos.
Are we happy with
Pero
looking further? Alright.
se funcionan.
Yeah. You want to come up on?
No, zero.
Bridge, that's auto view leaving bottom.
Leave in the bottom, okay.
All
Next
right.
slide.
You're welcome.
We have no reps.
Gracias para escuchar. Ahora subimos por casi 900 metros del columna de agua.
Look at that guy.
Vamos a apagar el audio en unos minutos.
Nice. Nice one, Paul. Happen? What's on the move?
Ahora estamos al cuarto de control. Juliana, Mike del Mediagua, Jess también del Mediagua, Laura, y muchos otros científicos. Gracias para tu interés y para escuchar. Thank you for listening. We have a couple more, 1 or 2 more dives to look forward to on this expedition. And we'll see you next time. Nos vemos la próxima emisión.